This is the video animated version of the Radwa and Radwa podcast, where we aim to communicate scientific research to science enthusiasts. We post 4 versions of each episode, in English and Arabic, and in podcast and YouTube video formats. All the links are in the description below. You will also find our references in the description. We are your hosts, Radwa Ewaisha,
and Radwa Sharaf.
PhDs, pharmacists, and molecular biologists. In our episode today, we will be discussing RNA vaccines. Radwa, could you please give us a recap from our last 2 episodes when we talked about DNA and RNA?
Sure. So 2 episodes ago, we started talking about DNA. And we said that within each cell in our body, there is a copy of our own DNA, and this copy needs to be protected from any potential damage or change in its sequence. And so whenever we need to synthesize a protein from one of the genes encoded in our DNA, we first need to make a copy of that gene in the form of a message, also known as a messenger RNA. And only this copy, this messenger RNA copy, gets exported outside of the nucleus so that the cell can synthesize the protein, which actually elicits a function inside the cell. So the DNA and the RNA themselves don't usually have functions inside of cells, but the protein does.
Okay, so because of COVID, everyone is talking about RNA vaccines. Could you give us a little bit of history and background about how the idea originated?
Sure. So the idea has been around for a while, almost 2 decades. Since we know that we can synthesize RNA in the lab, since its chemical structure is known, and if we know its sequence, we know which nucleotides need to be attached to one another to synthesize a message, scientists have been thinking about ways by which we can deliver that message inside of a cell so the cell can synthesize the protein encoded in this message. But the problem was, how do we deliver this messenger RNA that's synthesized in the lab inside of the cell so it makes the proteins encoded by it? And in 1987, a scientist realized that if the message is encapsulated with some droplets of fat, these fat molecules can bind to the cellular membrane that's also lipid-based. It's primarily made up of fats, and this allows for the transfer of the message inside of the cell, and so it can start producing proteins from it. And afterwards, multiple companies either originated or started developing mRNA vaccines for different types of viruses, but also for cancers, and they are in different stages of development.
Okay, can we talk about RNA vaccines against COVID? How do they work against SARS-CoV-2?
So the virus that causes COVID, SARS-CoV-2, encodes for 29 different proteins. When scientists started thinking about which protein to use in order to manufacture a vaccine, they picked the protein that's forming the outmost layer of the virus, which is called the spike protein. And so we can synthesize an mRNA molecule, so the message that will synthesize the spike protein inside of the cell. This message could either encode for the full spike protein or just a particular section or domain of the spike protein. And that's the approach both Moderna and Pfizer and BioNTech chose in order to synthesize a vaccine against SARS-CoV-2, with the idea being that once the mRNA is delivered inside of a human cell, this cell will start making the spike protein, and this will start stimulating our immune system to elicit an antibody and a cell-mediated immune response. This will be stored in the memory of our immune system, and so the next time our bodies are exposed to the virus because we were infected by SARS-CoV-2, our memory response can kick in and protect us. Now, it's worth noting, like I said, that it's challenging to deliver the messenger RNA molecule inside of the cell. That's why these vaccines relied on a technology that encapsulates these mRNA molecules with lipid nanoparticles that allow the mRNA molecule to pass through the cellular membrane inside of the cell.
Thanks so much for this. So, of all the misinformation that spread during COVID, the topic of RNA vaccines had a huge share. Could you please help clarify some of these?
Sure. So, around the time when RNA vaccines started development, a few misconceptions started circulating about the vaccine. So, let's tackle a few examples. One of them is that the mRNA vaccine can potentially alter or change our DNA or our genetic code. And that's not true because we just said that the mRNA messenger molecule is only delivered inside of the cell. It does not enter the nucleus. And it's actually quite challenging in the lab to deliver anything inside the nucleus of a cell. In order to do this, we have to add certain chemical compounds that allow for the delivery inside of the nucleus. So that's the first important point. mRNA does not enter the nucleus. But even if it somehow was able to enter the nucleus, which we just established it cannot do, but even if it did that, it still wouldn't be able to change our DNA because in order to change our DNA, um, we first have to administer compounds that break the DNA and allow for the external nucleic acid to integrate itself and bind to the DNA, which, like we said, is not something that's part of the vaccines that are being administered, and it's quite challenging to do this in the lab. The second point we wanted to clarify is that mRNA vaccines actually cannot persist inside the cell for a long time because, as we know, mRNA is a very fragile molecule. It can quickly be broken down and degraded into small nucleotides that are incapable of synthesizing a protein, which is the entity that elicits a function inside of the cell. And whether it's in the lab or in the form of these vaccines, mRNA needs to be kept at a very cold temperature, at least minus 20 degrees Celsius, to ensure that it doesn't break apart. And one final thing is that the mRNA vaccines, they don't contain any live virus or even any killed or inactivated virus. It simply has the message that encodes either part or the full length spike protein of our virus.
Thanks so much for the clarification. So, are RNA vaccines being developed for other conditions?
Yeah, there are multiple viruses for which mRNA vaccines are currently being tested. I would say the most advanced in terms of development so far is the mRNA vaccine against RSV, respiratory syncytial virus. There are a few companies that are testing out these vaccines in clinical trials, and they're showing quite good efficacy. There are also some mRNA vaccines against flu, as well as a range of different viruses, in addition to the development of vaccines against specific types of cancers. So I anticipate in the future we'll have many more mRNA vaccines that get approved over time. But so far, the only mRNA vaccine that is approved by the FDA is the one against SARS-CoV-2.
That's exciting. Thanks so much. So, to summarize, RNA vaccines have been in the pipeline for some time for several diseases before COVID hit. They work by allowing cells to create a foreign protein to allow its recognition by the immune system. RNA vaccines cannot change our genetic material and they do not persist for long. They are now being developed for several other diseases such as cancer and RSV.
We hope you enjoyed today's episode. Please don't forget to like and subscribe to our podcast and our YouTube channel. You can also find us on Twitter, Instagram, and Facebook. Feel free to reach out with suggestions for future episodes or leave us a comment directly on our YouTube video. See you next time!